Sorry. Let me know when you're ready. Yeah. All right. So, hey guys. So today we're going to be talking about the cell cycle control in animals and yeast. Oh. First off, let's reintroduce the cell cycle. So then the cell cycle, it starts at G1, then it goes into S, G2, and then it goes into M, which is mitosis. And we learned this last week, so it's kind of like a refresh. So what exactly is a cell cycle? So the cell cycle is a process in which the cells grow and divide into two new daughter cells. This is also known as interphase and is composed of three parts, like I mentioned before. G1, where it's preparing for replication, S, where DNA is replicated, and G2, where there's further growth right before it goes into mitosis. All right, so as I was saying, so G2 is right before it goes into division, which is mitosis, where the DNA and the cytoplasm is completely separated into two new daughter cells. So why exactly is the cell cycle important? So it's important because in eukaryotes, it allows them to repair their cells and tissues. And in the case for prokaryotes, it's important because it allows them to reproduce because that's their way of reproduction. So,
Right. So, as mentioned earlier, the cell cycle and its control system are incredibly important. For example, without this type of cell cycle regulation, cells may divide excessively, leading to cancer. On the other hand, if the cell cycle is not completed, new cells won't be able to replace damaged ones. In summation, the cell cycle control system is a set of molecules that sets the cell cycle in motion, regulating and coordinating its steps. It contains three major checkpoints: the G2, spindle, and G1. In general, the cell cycle control system requires a sort of timer or a clock that turns on each event at a specific time, allowing for the correct order of events to trigger only once per cycle. It also has a binary or on and off switch that triggers events in an irreversible fashion. Lastly, it needs to be robust and adaptable to compensate for malfunctions and changes in its environment. So, what is a CDC gene, otherwise known as a cell division cycle gene? CDC genes are essential genes that go for products required for the normal functioning of the cell cycle control system. It's so important that when CDC genes have mutations, the completion of cell mitotic activity is arrested at a specific point. This is also the reason that these mutated CDC genes cannot be propagated normally for studies or for research. Instead, to work around this obstacle, researchers created something called a conditional cell cycle mutant. The majority of these are temperature sensitive. For example, here we can see the permissive low temperature completing its cycle, while the restrictive high temperature arrests in the G1 phase. This allows for these mutants to not only be maintained, but also for their effects to be noted and studied in many fields, including gene therapy, where mutated genes are knocked out and replaced with healthy ones. All right, next we're going to talk about CDKs, or cyclin-dependent kinases, which are a family of enzymes that modify protein substrates involved in cell cycle progression. Specifically, this is done through a phosphorylation mechanism. It is also important to note that different types of CDKs exist, with many being found in animal eukaryotic cells and only one being found in yeast cells, which we'll go over in a bit. As it suggests in the name, CDKs require conformational change to be activated by the binding of cyclin. Cyclins, on the other hand, are a family of proteins that mostly exist to activate CDKs. However, they do play a role in directing them to specific target proteins. Cyclin, however, degrades throughout the cell cycle. This is done through regulated proteolysis and a ubiquitin mechanism. This degradation is important because as the cyclin levels decrease, CDKs slowly become inactive. Essentially, it acts as a sort of on or off switch, like we discussed earlier. If cyclin does not degrade, cell cycle arrest will occur. So here I'm going to go over a brief example of a ubiquitin mechanism and protein degradation. So you can see in the image, ubiquitin is transferred from E1 to E2. E3 then recruits the substrate transferring. Ubiquitin is then sent to E2 and the substrate. Finally, it is able to go through protein degradation.
So since we talked a little bit about CDKs and cyclins, let's talk about them in animals and yeast. So like mentioned before, animals have 4 CDKs. They have CDK4, 6, 1, and 2. And these CDKs actually interact with cyclins, right? So then 2 of them interact with cyclin G1 cyclin, one with G1 slash S and S cyclins, and one with S and M cyclins. So specifically, CDK4 and 6 control the G1 to S transition. And they do this by activating DNA synthesis. Without activating these CDKs, S phase cannot begin. So the whole cell cycle is stalled. So in comparison to yeast, they only have one CDK, CDK1. So this one CDK is very versatile in that it can bind to all classes of cyclin. However, CDK1 binding to a specific cyclin doesn't exactly trigger the same interactions, but it triggers different interactions depending on what partner cyclin or CDK1 binds to. And interestingly enough, a human homologue of CDK1 can also be used in yeast and function completely normal. And for this, we can assume that the cell cycle and its mechanisms are probably conserved between animals and our humans and yeast. As you can see, a lot of the cell cycle is regulated by checkpoints, which we'll discuss in the next slide.
All right, so here we have the checkpoints. The checkpoints are points in the cell cycle that examine not only internal, but also external cues, deciding whether or not to continue the cycle. The G1 checkpoint, found between the G1 and S transition, asks the main question, to divide or not to divide, and checks to see if the cell is the correct size, has enough nutrients to divide, the correct growth factors, as well as checking for DNA damage. The G2 checkpoint, found between the G2 and M transition, asks the questions if DNA has enough integrity and if it has replicated correctly. But what happens if it finds that the integrity is compromised or the DNA is damaged without repair? It can send the cell through programmed cell death, aka apoptosis. The last checkpoint is the spindle checkpoint, and this one does not scan for metaphase plate and its corrections. Instead, it looks for straggler or misplaced chromosomes. If one is found, the cell will cause a delay until the spindle can capture the stray chromosome and repair the cell.
So how are these checkpoints controlled? So these checkpoints have protein interactions that include CDKs, cyclins, and CKIs, which help with the transitions within the cell cycle control. So let's go over some examples. So for the first example, CDK is low in G1 phase. However, when there's a rise in S cyclins, there's also an increase in CDK activity. This increase in activity triggers the G1 to S transition, activating the DNA replication. Here's another example, example 2. So the example 2 can be seen in the photo, but in example 2, at the G2 checkpoint, if DNA is damaged, ATM kinase activates CHK1 kinase, inhibiting CDC25, which blocks the activation of mCDK, which stalls the whole cell from going into mitosis. And as you can see, it's kind of like a cascade, or like everything happens after another. So now, since we know about the cell cycle and how it's regulated by the cell cycle control, let's also talk about them in animals and yeast again. So for animals, proliferation, how the cell grows, is ultimately dependent on cell signaling and how the organism itself grows. This process is very tightly controlled. If the control was to be lost or unregulated, it could lead to cancer. The presence of mitogens, which are small proteins, induce a cell to begin cell division and helps stimulate its growth. During the process of M phase, the nuclear envelope is broken down and microtubule spindles form, specifically in prophase. And now, in comparison with yeast, proliferation is dependent on nutrients availability, making it super fast if there's a lot of nutrients available. With this, there's also a size is the controlling factor, because in yeast, since it's growing dependent on the nutrients, it needs to be able to achieve a certain size before continuing. So, and in the case of yeast, during the M phase, there's no breakdown of the nuclear envelope, and microtubules form inside the nucleus in the early S phase. And here's some websites we used, including the book and lectures. And that's our presentation. Any questions?
So essentially, because cancer is the like excessive cell division, the cell cycle control helps control that to where it's not just continuous cell division. It helps regulate it essentially.
With the checkpoints.
For example, how we talked about like the on or off switch or the timer, those are seen in the molecular transitions. So essentially, it would be able to pause the cell if it was, or delay the cell if it found there's errors in replication, or too much replication in this case.
Can you go back to your examples?
Yeah, give me a second. I need to share this screen again.
No, I'm group number 4.
These examples?
Yeah, can you can you explain more of example 1? Because how is like low CDK activity in G1, and then there's a rise of CDK? Like that's a little confusing to me.
From what I understood, that since in the beginning, when it's in G1, it's just in the process of growing. CDK is not as high, because CDK and cyclin helps with the progression and growth and division of the cell cycle, right? Or the cell. So then as it keeps on moving, the activity increases with the S cyclins, which allows it to, move through G1 to S, if that makes sense.
Yeah, okay, thank you.
Mhm.
Sorry, I had a question. I was oh, what? Oh, I'm number 7. I was wondering if you could go back to the how CDK is regulated in.
Could you say that again? I
Oh,
can't
sorry.
hear
I
you.
was wondering if you could go back to how CDK is regulated in yeast cells.
Regulated in yeast cells?
Yeah.
Wait, this one?
Go back a bit more.
Ba-
Give me the
is it this
diagram.
one?
Yeah. Yeah.
Okay.
I was wondering if you could explain like a quick summary of how it's regulated because I remember you saying that it's just one general CDK molecule and how it
I'm sorry,
regulates.
I can't really hear you.
Can you hear me now?
Yeah,
Oh,
sorry.
no, it's okay. I didn't know where my microphone was on my laptop.
It's
I
alright.
was wondering if you could re-explain how that one CDK molecule basically, I guess, controls everything and how it's regulated
Oh,
compared to animal CDK.
okay, so then obviously with the animal CDKs, we're larger organisms, so then we're gonna have a little bit more stuff, we're a little extra. So then in yeast, they're just singular celled organisms, so then obviously they're gonna have one CDK. And you're asking how that one CDK is able to bind to all classes, correct?
Yeah.
Well, I'm assuming since it's a single organism, it still needs to be able to do what an animal can as well, or like a multicellular organism could do. So then it has like the ability to do the same, but with less, if that makes sense.
Yeah. Okay. Thank you.
Mm-hmm.
I also had another quick question.
Yeah.
So, if you restrict the CDK4 or the CDK2 or both of them, what would really happen to the cell cycle, especially the G1 to S phase, and how would that affect the DNA synthesis?
Okay, so for your first question, if the CDK4 and 2 were like mutated, I'm assuming? Like, could you repeat your question?
Well, I was wondering if you either like restrict those enzymes, like the
Mm-hmm.
CDK4 or CDK2, or mutate them somehow so that it doesn't maintain its function to
Mm-hmm.
control the G1 to S phase, what would happen to the DNA synthesis?
Well, I'm assuming that since the CDKs help regulate the cell cycle control, if it were to be like misregulated or like mutated, it will go back to the G0 phase and then wait for everything to, be all good.
Essentially, the G1 checkpoint would be like, all right, something's on right here. I would create a pause or delay until it could gather them, or if it's or it would go through apoptosis if it can't be fixed.
Gotcha. Thank you, guys.
Yeah.
Okay. I think so. Okay.
Okay. So our group will be discussing mutational experiments analyzing the cell cycle. Next slide. So there are 3 key experiments that show how the eukaryotic cell cycle is regulated. The first set of experiments are cell fusion experiments, which show the presence of a maturation promoting factor, which is a factor that drives cells to M phase or mitosis. The second, yes. And the second set of experiments are the oocyte experiments, in which the biochemical analysis of mature oocytes show that they contain MPF and the proteins that are associated with this factor. And lastly, the third set of experiments involve the genetic analysis of temperature-sensitive yeast mutants, which help to identify specific proteins that are involved in cell cycle transitions.
So we're going to talk about cell fusion experiment. So cell fusion is an important cellular process in which cells with a single nucleus combine to form a multinuclear cell. So similar to what was said on the previous slide about cell fusion, there's a presence of MPF and the presence of an S-phase clonal factor. As you can see, there are 2 cells presented in this image. The mitotic cell is in cell division, and it is going through mitosis. With cell fusion, the mitotic cell infuses with a cell that is held in G1 phase. What happens in cell fusion is that in an induced cell, the DNA that's in the G1 phase cell will start condensing and make chromatin. This condensation of the chromatin is then assigned to enter the M phase, which is also known to be the mitotic phase. This then suggests that the mitotic cell contains some sort of soluble factor that is responsible for driving the G1 phase cell into M phase because it is being used in the condensation of chromosome in the G1 phase. So to start off, DNA replication begins at origins of replication, which are scattered at various locations in the chromosome. In this diagram, you can see that in late mitosis and early G1, proteins bind to the ORC at origins. The ORC is an origin recognition complex. It is a multi-protein complex that binds to the yeast replication origin. It is the start of DNA replication and serves as landing pads for several additional regulatory proteins. In the G1 phase, the proteins being used here are CDC6 and CDT1. Right after, you can see that another protein appears. G1 recruits one more protein called MCM, which is known to be a helicase. The function of this MCM helicase is to bind the protein so it can become the pre-replicative complex. Once the SCK triggers the S phase, which means that it will trigger the degradation phosphorylated CDC6. After that, it is followed by the inhibition of CET1 by geminin. SCDK, which is cyclin-dependent kinases, then recruits the DNA polymerase and other proteins to form the pre-initiation complex. SCDK, with help of additional kinase, collaborate to phosphorylate the origin recognition complex. And then next, MCM helicase is activated to unwind the DNA and initiate replication. This will begin the synthesis of the DNA.
So next we'll talk about the oocyte mutation experiments. So this experiment was conducted by Yoshihiro Masui. and it involves the use of synchronized cell division, where the cytoplasm of this unfertilized egg, which is shown in the left, with um, the during multiple stages in embryonic development. As you can see here, it's in metaphase, and into an immature oocyte egg, which is in the G2 phase of development. And it was found that as a result, spindle formation occurred in the oocyte egg, which is shown by this green egg over here, and after the injection of the cytoplasm. And this showed that there is a maturation promoting factor in the cytoplasm that was injected into the immature egg, and this basically drove the G2, the phase immature oocyte that was in G2 into the M phase, which is mitosis. So next, let's talk about the maturation assay. And first, I want to talk about this figure. This figure shows that the MPF activity is high during embryonic mitosis, and it shows an oscillation pattern of MPF activity during the synchronous embryonic divisions. And as you can see, the MPF activity is high during the first embryonic mitosis and the second embryonic mitosis right here. And from each stage of this embryonic mitosis, Masui extracted proteins and injected them into the immature oocyte egg, and used them as a maturation assay to see whether the oocyte eggs would go into the M phase.
Okay, so next we're going to be talking about MPF purification. This was achieved by running 6 different chromatographs using the cytoplasm that was extracted from the fertilized oocytes. The chromatographs revealed that MPF consists of 2 important polypeptides, P32 and P45. P32 is a protein kinase that helps in phosphorylating P45, which is a histone. It can also be noted that P45 alone is sufficient enough to promote metaphase in a cell, which can imply that P32 plays a role in the pathway of MPF action and is responsible for inducing mitosis. So, cyclin discovery. Cyclins are a group of proteins that control the cell cycle by activating CDK enzymes. This group of proteins undergoes specific protein synthesis that is needed for the cell to enter mitosis. They were discovered during an experiment that utilizes sea urchin embryos. During this experiment, 35S-methionine was added to the embryos, and then they were fertilized. Protein extracts were then sorry. Sorry, I got a little nervous, I'm sorry. Protein extracts were then made from these fertilized eggs and then run on an SDS page. Sorry, sorry, I get really nervous during presentations, I'm really sorry. Sorry. So during the experiment, 35S methionine was from these fertilized eggs.
Okay, so lastly, let's talk about the temperature-sensitive mutation experiment. This experiment involves divisions mutants in yeast, of which 2 types are analyzed: Saccharomyces and Schistosaccharomyces pombe, which is a fission yeast used to produce African beer. So on this diagram here on the right is the cell cycle progression of the 2 different yeasts, as mentioned before. In fission yeast, the cell elongates by growing at the 2 ends, and these 2 ends grow, leading to the elongation of these large-shaped cells. And once they reach a particular size, they undergo mitosis. In budding yeast, cell division occurs through the formation of a bud. The bud is formed during G1, which is the starting point, and the cell commits to cell division, followed by the bud growing due to this aligned cytoskeleton, which ultimately leads to the separation of the bud through cytokinesis. So as you can see, the size control point in budding yeast is between G1 and S, whereas in fission yeast, the size control is between G2 and M. Next slide. Next slide. Oh, back one more. Okay. So these CDC mutants have mutations which cause cells to arrest or stop at a specific point in the cell cycle, meaning a normal gene product is required to get past this point for proper propagation to occur. So in most conditional cell cycle mutations are temperature-sensitive mutations in which the mutant protein fails to function at high temperatures, but functions well enough at low temperatures for cell division to occur. A temperature-sensitive CDC mutant can be propagated at a low temperature, which is also known as the permissive condition, and then raised to a higher temperature, which is known as the restrictive condition, in order to turn off the function of the mutant gene. As you can see in the following diagram, it shows various CDC mutants defective for cell cycle transitions. In the first example, CDC28 mutants undergo proper cell division at permissive temperatures, and at restrictive temperatures, they fail to make a bud, which means that the arrest point was between the start point or between G1 and S for this particular mutation. In fission yeasts, such as CDC2 and CDC13 and CDC25 mutants, they fail to divide after reaching a particular size, so that means that the division or arrest point is between G2 and M and indicates that these cells have a problem in making a G2 to M transition. At the same time, another mutant called W1 was identified. Instead of making regular size cells, they started making extremely small cells, which means that there was an unrestricted entry of these cells in G2 to M transition. Next slide. So once you can identify these mutations, it's easier to clone the genes in yeast. One general strategy that is commonly used is complementation cloning, which involves taking a mutation and transforming it into a library of yeast DNA fragments, which represents all the possible genes in yeast. And then once you go back and look at which one of these clones rescued the particular mutant, it suggests that the particular gene responsible for creating this particular mutant phenotype is it's responsible for creating this particular mutant type. So in that way, you can go back and sequence the gene and then identify the protein product responsible for making this transition. So that concludes our presentation. Any questions? So MPF is a maturation promoting factor, the mitosis phase.
It promotes mitosis. It essentially takes the cell that's in the G2 phase and, or is it G? Yeah. And just, it basically signals it to start dividing. This one? Um, shoot. Um, is Amy here? Or like, what do you mean by the question? Yeah. Mm-hmm. Basically,
Is it that the mitotic cell contains a factor that drives the G1 phase into the M phase? Okay.
is it this one?
So basically, what they found out was that when the cytoplasm was injected from the unfertilized egg in the metaphase into the immature oocyte, in G2, it basically they saw formation of spindles, right? And they basically discovered that the cytoplasm of this egg in metaphase contained a maturation promoting factor that drove this immature oocyte into the M phase, right here, or mitosis.
Yeah, we have a question about the Tim Hunt cyclin discovery experiment from Group 5.
Okay.
We were wondering if, like, why particularly sea urchins were used for the experiment.
I don't think there was a particular reason. I don't remember seeing a reason, to be quite honest. But it could just be that they're easy to, I guess, breed, or the embryos were easy to use, I guess.
Okay, thank you.
Mm-hmm. Okay.
Yeah, I have a question. I'm from Group 9. Um, so I was wondering what the difference is between CSF and MPF.
Yes. CSF?
Yes. Am I mistaking that for something else?
I don't think there's a CSF here. No.
No.
Okay, sorry guys, I think I misspelled it.
S.
That's okay.
It's an S. We have MPF and SPF. Yeah.
Okay, gotcha.
In like MPF is for like the M phase and SPF is probably for the S phase.
Mhm.
Got it. Thank you guys.
I have a question.
Any other questions?
Can you go in? Can you explain the chart where there's different in the experiment where from the sea urchin? Yeah, can you explain what the graph says?
It pretty much just had to do with how the proteins over time, how they peaked during certain events in mitosis.
Right, thank you.
Or throughout the cell cycle. I'm sorry, not mitosis.
I asked a question, like a clarified question. When we were talking about the oocytes, about the egg.
Mhm.
Did you guys say that it shows a nitrogenetic factor in the egg?
It shows a maturation promoting factor in the cytoplasm of the unfertilized egg, which is then injected into oocyte, and then that drives the G2 phase to mitosis.
No, I can't. Yeah.
I think we're ready. I'll just share my screen. Okay, can you guys see that?
Yeah.
Yeah.
Okay. Let me know when I can start, Albert. Okay. So my group is going to be talking about cyclins and CDKs. So I noticed that a couple of the groups before us, our topics kind of overlap. So hopefully we'll be able to give you guys a little bit more insight on the cyclins and CDKs. So our topics to be covered, this is a list of what we'll be covering. I'll kind of give you guys like a general overview of what cyclins and CDKs are, and then my group members will go into a little bit more detail on the mechanisms of how they work and what they do. So cell cycle washing machine. So I thought a good analogy for the cell cycle was kind of like a washing machine in the sense that it goes through phases just like the cell cycle. You have your pre-wash phase, the washing phase, the rinsing phase, the spinning phase. And in the same way, the cell cycle has a G1 phase, the S phase, the G2 phase, and the M phase. So just like a washing machine, it needs to be regulated. You don't want to put your clothes in and set it to the pre-wash phase and have the dial turn all the way to the spinning phase before your clothes are washed. And so the mechanism for a washing machine is that as In the pre-wash phase, as the washing machine fills up with water, there's like some sort of pressure. There's like a pressure type of mechanism where it senses how much pressure is in the tank, and that lets the washing machine know when it's full to start the washing cycle. So in the same way, the cell cycle has a mechanism to tell it when to continue or if it needs to stop. And so we will discuss what that mechanism is and how it works. So they control the cell cycle through cyclins and CDKs, and cyclins are a group of proteins that control the progression of the cell cycle or of a cell through the cell cycle. So there are four main classes of cyclins: G1 cyclins, G1S cyclins, S cyclins, and M cyclins. And these are each named based off of the phase in the cell cycle that they bind CDKs and they're most active. So the G1 cyclins will bind CDKs and are mainly active in the G1 phase, but as you can see from the graph, they kind of are constant throughout. And this is because G1 cyclins are kind of like regulators for the G1S cyclin and the S cyclin. And then G1S cyclins are mainly active in the G1 late G1 phase to the S phase, and then S cyclins are mainly active in the S phase and M in the M phase. I think that's it. This one. So, what are CDKs? So CDKs are cyclin-dependent kinases. They're a group of protein kinases that will modify different protein substrates throughout the cell cycle. So these are the actual activators of the events that take place, such as dividing the DNA, splitting the cell, things like that. So CDKs are not active unless they're bound with cyclin. This graph is just here to show you that the concentration of CDK is constant throughout the entire cell cycle, and cyclin varies, which is the reason why they actually called it cyclin, was because the concentration varied throughout the cycle in a cyclic nature, and so they call them cyclins. Like I said, the concentration of CDK is constant throughout the cell cycle. And this is mainly because the process of cell division needs to happen quickly. So you don't want to have to make CDK just as soon as you get to the phase that you're trying to complete or go through. So you want to have this stuff kind of ready. And so CDK is always active, but because it's not always active, but it's always in a high concentration. But because of this, the cell has to have a way to regulate this. So CDK interactions. So basically what happens is CDKs will bind with cyclins and they'll have interactions throughout the cell cycle. So this chart is just a chart of different CDKs and their cyclins and what phase of the cell cycle they function in. So CDK regulation. So like I mentioned before, CDKs have to be regulated because they're in a high concentration. So they always need to, the cell needs to have some type of control over the CDK so that they're not going and phosphorylating things and activating things ahead of time. Because we know the result of that is usually like cancer, tumors, or something like uncontrolled cell growth. So the first layer of regulation is the binding of cyclin itself. And so you can see from this schematic that a CDK that's not bound to a cyclin is inactive. And once it binds to a cyclin, it's partly active. So it isn't active yet, but it's kind of prepped for when the cell gets to that phase. And the second layer of regulation is mediated by other protein kinases and phosphatases. So the important proteins are Wee1 kinase, CAC, which is not shown in this schematic, and CDC25 phosphatase. So what happens is once the CDK binds with cyclin, WEE1 kinase and CAC will phosphorylate the CDK cyclin complex in two different places. So CAC will phosphorylate it in this activating site here, and WEE1 kinase will phosphorylate it in the inhibitory site over here. And so what will happen is once the cell reaches the phase where it's about to transition into another cycle, this CDC25 phosphatase will dephosphorylate this inhibitory phosphate, and this will allow the CDK cyclin complex to become active, and it can phosphorylate and activate whatever it needs to progress the cell cycle. So another layer of regulation is CDK inhibitor proteins, or CDKIs. These will bind to the cyclin CDK complex and cause a conformational change that will inhibit the whole complex. So these are usually used in the checkpoint mechanisms, which I'll talk about a little bit later. So these will bind and prevent the cell cycle. So these will basically arrest the cell cycle at a certain point and prevent the cell from progressing into the next phase. So the checkpoints. So like I mentioned before, the cell has to regulate how the cell grows and moves before it divides. So it makes sure that everything is correct. Kind of like a washing machine. The washing machine has to regulate when there's enough water in the tank, if it can move on to the wash phase, and it does that by its pressure gradient. And so it's the same way with the cell. The cell will check at certain points in the cell cycle and make sure that everything is correct before it moves on to the next phase. So the major checkpoint phases are the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. And I'll go into a little bit of detail for all three of those. So the G1 checkpoint basically checks if there's enough nutrients, if the size of the cell is okay, if there's enough growth factors for the cell to continue growing in later phases. And the most important checkpoint in the G1 checkpoint is DNA damage. So if there happens to be DNA damage at the end of G1 or during G1, because the cell is continually checking, the cell has a mechanism basically to find if there is any DNA damage and then try and repair it. So the important proteins for this mechanism are P53, P2, P21, and MDM2. So the schematic here is showing the DNA is damaged by like X-rays. And then you have P53 bound with MDM2. So normally P53 is always in the cytosol at very low concentrations. It's always around and it just gets degraded. So if there's no damage in the DNA, P53 will just get degraded. It'll be bound to MDM2, which will, I guess, signal it to be ubiquinated, and it will just be degraded. If there happens to be DNA damage, it'll increase the amount of P53 and activate the P53. And so P53 is a gene regulator. And so once it's activated, it will regulate the transcription of the other protein, P21, which will code for the P21 CDK inhibitor protein. And this is what will bind to in this case, the G1S CDK and S CDK and prevent them from progressing the cell cycle into the S phase. So this protein will stop or arrest the cycle until the DNA is fixed. And if it can't be fixed, like I think the first group mentioned, then the DNA will, or the cell will end up going through apoptosis and it'll die. The next checkpoint is the G2 checkpoint. So in this checkpoint, the cell is checking if the DNA was replicated correctly in the S phase. it'll check again for DNA damage, and it'll check if there's enough proteins to continue to grow and divide. The most important check in this point is for replicated DNA. I wasn't able to find the schematic for this, but basically, I'm assuming it's similar to the DNA damage checkpoint, where if it's damaged, there's a mechanism to try to repair it. So in this case, I guess it's not replicated correctly. I'm assuming that there's some type of mechanism to either go back and re-replicate or something like that. The last and final major checkpoint is the M checkpoint. So in this checkpoint, we're seeing if all the chromosomes are attached to the spindle, if spindle fibers have formed, and if the chromosomes are oriented correctly. So in this checkpoint, the major thing is spindle assembly. So you can see here in this schematic, there's two different scenarios. So when the checkpoint is on and active, this is basically something went wrong. The spindle fibers did not connect to the kinetochores on the chromosomes. And so what will happen is the checkpoint will turn on, and there will be a mechanism that will prevent basically the splitting of or the separation of the chromosomes. So my group members will talk a little bit more about some of the proteins involved in this. And then when the checkpoint is off, that means everything happened correctly. So the cell is saying, okay, check, everything is connected correctly, the chromosomes are aligned. So we can go ahead and allow the cell to go ahead and divide and split the chromosomes. And that's what you see here. So those are the major checkpoints of the cell cycle.
Can you guys hear me?
Yeah.
Okay, so I'm gonna be talking about how CDK is activated. So like Brandon said, CDK helps regulate the progression of the cell cycle. And in each cell cycle, there's different types of CDKs that associate with different types of cyclin to help progress and activate molecules. So in the absence of cyclin, CDK remains inactive because the ATP active site is hidden. And then in the presence of cyclin, there is a conformational change in its shape that allows the ATP active site to be exposed. And that is when it's not partially, like, it's only partially active. It's not like fully active yet until CDK activating kinase comes in and phosphorylates an amino acid near the entrance site of the active site that increases the activity of CDK. And as you can see in the last structure, CDK becomes fully active. Next, please. Okay, so we're going to be talking about mitogens now. So they are one of the extracellular signal molecules that allow cells to like commit into the cycle. And they're made of it's a peptide that induces a cell to begin in cell division. And so as you can see in the picture, when mitogens are activated, they allow cyclin D levels to increase, which binds to CDKs and become activated. Whereas in the absence of mitogens, cyclin D levels are like too low to bind to CDKs. So the cell enters a G0 phase, and so it doesn't commit into the cell cycle. And if you look in that picture, there's an R point, which is known as a restriction point, where once a cell is at the restriction point, it no longer requires, is no longer dependent on mitogens for further proliferation. Next. Next, please. So, oh, can you go back one slide? So before I go into depth about this model, I want to talk about the different types of proteins that are involved. So one of the proteins is called retinoblastoma protein, which is a tumor suppressor protein that restricts the cell's ability to replicate DNA, and so prevents like the regression from G1 to S phase. So basically, mitogens allow cells to commit, whereas RB proteins prevent it from being committed. And another molecule would be it's called E2F, which are transcription factors that pushes cells into the S phase, and it transcribes like CDKs for further cell synthesis. And so mitogens basically stimulate the G1 CDKs and G1S CDKs. And as you can see in this picture, they trigger a series of like steps that activates G1 CDK, which then phosphorylates the RB protein to inactivate it. So when RB is inactivated, E2F is basically free and exposed to translate for like different types of CDKs like G1 and G1S. Next, please. So APCs are known as anaphase promoting complex. So they basically have two overall functions in the cell cycle. One is to trigger the transition from metaphase to anaphase. So as you can see in this picture, so separase is a kind of enzyme. It's a type of like protease that cleaves the cohesin, which binds the mitotic spindles together. And so when APC is activated, it basically activates the separase, and then the separase like degrades it cleaves the cohesin from the chromatids, and then which allows the chromatids to like split, like go to the opposite ends of the poles, which triggers anaphase. And so right when the cell enters anaphase, it needs the M cyclin needs to be cleaved right away in order for it to continue onto the next cell cycle, and so which is mitophase. And then so the role of APC is it controls like cyclical proteolysis, where it binds. So basically, it's inactive until CDC20 binds to it, which then recruits ubiquitin like proteins that binds to mCDK and triggers the breakdown of M cyclins. Yeah.
Hello? Sorry. Can you guys hear me now?
So like it's very quiet.
Okay, let me try to. This one is working actually. You guys can't hear me?
Like it's very
super low.
Okay, let me try. How about now?
Yes.
Okay, yeah. So, thank God. So I'm going to talk about the cyclical proteolysis and how cell cycle control depends on it. Most cyclic components are activated by regulated proteolysis of a cyclin at a certain stage. This cyclin distraction occurs by ubiquitin-dependent mechanism. Ubiquitin is a tag that marks the protein for proteasome degradation. So this is the mechanism for the ubiquitin proteasome system. And how it works is basically the ubiquitin is like initially added to the So this mechanism leads to like degradation of a cyclin once a cell gone to like one particular transition. So how it works is the ubiquitin is like initially added to the like a specific enzyme, the E1, the light blue one, and the E1 transfers the ubiquitin to like the E2 enzyme, and with the help from the E3 enzyme, the ubiquitin is transferred to like a substrate cyclin, like to a substrate, and we form this cyclin right here, and it's sent to like degradation through the proteasome mediated degradation process. Can you go to the next slide? So those are like the two types of ubiquitin complexes that ubiquitin targets. We have control of proteolysis by SCF and the control proteolysis by APC. So how each works is like basically the first one, the one on the right, works is like the APC is activated in mitosis by associating with the activating subunits, the CDC20. And like this subunit, the CDC20 recognizes a specific amino acid sequences on the M cyclin, and with the help of those two like enzymes right here, like the APC transfers a multiple ubiquitin molecules to the targeted protein, and then the I'm not sure if I'm saying right, but the polyubiquitin target recognizes and aggregates the proteasome. And like it's the same, like they're both, I think they're both similar, but this one is like the one by SCF is dependent on the F box protein. So for the second one, it's like it depends on the substrate binding subunits, which is called the F box, and the phosphorylation of a target protein, like such as the CKI, allows the target to recognize that by to be recognized by the F box. Then like same as the first one, like with the help of those two enzymes, the ubiquitin are like being like transferred, like the molecules are being transferred. Then we form and it recognizes like and degradation. I'm sorry. Then the polyubiquitin target is recognized and the degradation in the proteasome. Can you go to the next slide? The intercellular controls. Each cyclin CDK serves as a molecular switch that triggers a specific cell cycle event. And I think here we're trying to understand how the switches initiate such events in a correct order and once per cycle. Can you go to the next one? So like first I'm going to talk about the G phase. This is like this phase, like as it mentioned before, like basically in this it's like in this phase is like we have like the end of the cyclosynthesis and the start of the DNA synthesis. Here like the cell growth and the cell cycle progression is monitored by the total amount of the G1 cyclin, which is also called the CLN3. The CLN3 is synthesized in a parallel with the cell growth and its concentration, and it always remains constant. And if it's artificially increased in a cell, it will divide. The cell divides. I mean, this mechanism is not like well understood, but the one possible way to explain it is like a cell may have a fixed amount of inhibitors of the CLN3, but if it exceeds the extra amount triggers the G cyclin activation and a new cycle. Since all cells receive the same amount of DNA, it has been like speculated that the DNA was in the inhibitors of the CLN3. And the S phase. So in this phase, basically like the DNA synthesis occurs. So this like initiates the DNA replication once per cycle. It's like it prevents re-replication and not only minimize the risk of mutation, but also prevents a damaging gene. So it's a cell that have completed like the S phase are not able to replicate their DNA, even if when they're provided with the SCDK activity. And a passage through mitosis is required for a cell to regain the ability to undergo S phase. So this is the cell fusion experiment for replication block. So on the first one, on figure A, we have the G1 cell fused with the S phase cell and DNA replication initiated. Like as a result, we have the DNA replication was initiated in the G1 nuclei. And for the second one, we have the G2 cell fused with S phase cell, and like there is no synthesis. As a result, we don't have any synthesis. Like there's no synthesis. And for the third one, we have the G1 fused with the G2, and yeah, it's like I don't know how to like find this, but it's like according to lectures and everything, it says the G2 stays in the G2 and the G1 like enters the S phase as expected. So this is like two more, like this one and the last one. Okay, so the molecular basis of a cell fusion. I think this was like mentioned before. So here, the DNA replication begins at the origin of replication, scattered at the various location of the chromosome. So in this mechanism, we have in this experiment, we have the G1 and the CD6 and the CD1 associates with the origin recognition complex, which you basically serves the ORC basically serves as a landing pads for several additional regulatory proteins. And then on the second stage, we have the MCM helix ring complex assemble on the flanks to yield to pre-replicative complex. Then we have this SCDK recruits the DNA polymerase and other proteins to form the pre-initiation complex. And on the third one, we have then the MCM activated to unwind the DNA and initiate replication. And finally, the SCDK triggers the CD6 distraction and the OR formation. The Gemini inactivates the CD21. And the last phase is the M phase. So in this one, basically the cell division occurs. So here we have a diverse and a complex rearrangement that occurs in early stage of mitosis and induce the assembly of mitotic spindle and ensures that each sister chromatides are attached to the opposite pole of the spindle, and it triggers the chromosome condension. Also the envelope breakdown. Also it also triggers the actin cytoskeleton and the rearrangement triggers the actin cytoskeleton rearrangement and reorganization of the Golgi and the ER. And finally, the MCDK phosphorylation, several proteins that regulate the microtubulous behavior. So this is the mechanism how to activate the MCDK mechanism. So basically, in this mechanism, we have the CDK1 associated with the M cyclin, and the resulting MCDK complex is phosphorylated on activating site by the CDK activating kinase. Is that how you say it? And on a pair of inhibitory sites by the V kinase, and the resulting inactive CDK complex is then activated at the end of the G2 phase by phosphate CDC25. And the CDC25 further stimulated by active MCDK, resulting in the positive feedback. This is the end of our presentation. Do you guys have any questions?
One second.
Okay, are you ready?
Yep.
Okay. Apoptosis, extrinsic and intrinsic. What is apoptosis? It's basically death of a cell, which occurs as a part of an organism's growth or development. It's an orchestrated process that results in an organized degradation of cells. It leads to the elimination of cells without releasing harmful substances into the surrounding area. Not to mention that it's key to maintaining our health as we are getting rid of unnecessary cells. Although apoptosis is important, too little or too much of it can play a role in many diseases such as cancer, Parkinson's, or Alzheimer's. Why does it happen? There's various reasons why apoptosis may occur, one of them being that cells can be severely damaged to the point where they cannot be repaired, so the easiest way to fix the problem is to die. There also could be that they're no longer useful, they have served their purpose, and it's time for them to leave the organism. Another factor is that cells get old and they don't function properly, so when it's their time, they just die. And lastly, apoptosis plays a key role in development, such as they could help develop fingers in a baby during embryonic development.
Okay, so we will now discuss the molecular basis behind apoptosis. And it essentially starts with the caspase cascade. A caspase is pretty much an intracellular protease that cuts up essential proteins found throughout the cell, which will essentially just which will result in an organized cell death. The caspase name is derived from key amino acids found within that protein. For example, C standing for cysteine, which is the amino acid found in its active site, as well as aspartate, which is the target amino acid that is cleaved during apoptosis. This cascade is irreversible, meaning that once it starts, it cannot turn back. And there are two major classes of caspases: the initiator caspase and the executioner caspase. This cascade first starts with the initiator. It's first inactive and requires two isolated and individual monomers of initiators. These inactive monomers bind to an adapter protein, which will activate it via dimerization. And the two monomers combine via cleavage, and that becomes an active initiator caspase. Downstream, you see that the executioner caspase, which is by its name the protein that is actually doing the cutting, is activated by the initiator caspase. The apoptotic signal that initiates this entire process could be derived from either outside or within the cell, either through the intrinsic or extrinsic pathway. We'll first talk about the extrinsic pathway, which is pretty much the target cell receiving an external signal from either an immune response via these lymphocytes or white blood cells. Right, and so one well-known example is the FAS protein. So this killer lymphocyte, as you can see in this schematic, binds to the FAS death receptor of the target cell that will be programmed to death. And so, sorry about that. And so the fast ligand and the death receptor will combine, and this death receptor has three different domains. It has the ligand binding domain, the transmembrane domain, as well as the intracellular domain that is found within the cytosol of the target cell. And pretty much once they bind, it initiates a conformational change within the cytosol that forms this disc, which is the death-inducing signaling complex. And at the bottom of this complex are inactive initiator caspases. And so they, once this disc is formed, they activate these initiators and they cleave off on their own to activate and initiate the caspase cascade that I mentioned in the previous slide. It's all really similar. It's essentially the same pathway that induces apoptosis. And on the other side, as we have the intrinsic pathway, which is the trigger of apoptosis via signals found within the cell. Some of these signals could be derived from lack of oxygen or nutrients, DNA damage, or developmental signals. One well-known example is the release of cytochrome C, released from the inner membrane space of the mitochondria. This cytochrome C, which while it plays a key role in the electron transport chain of the mitochondria, sometimes it does get released because of these stresses. And so it will bind to an adapter protein called APAF1. And this combination will will then polymerize and form a wheel-like structure called a apoptosome. This apoptosome has a center ring full of cars, which are caspases recruitment domains. And so you have surrounding caspase, initiator caspases in the surrounding cytosol. And so they will all be recruited onto the apoptosome and essentially activate it and induce the caspase cascade that I mentioned on the previous slide. And so it's very similar, pretty much rinse and repeat. That's just how you receive that apoptotic signal to begin apoptosis.
The intrinsic pathway is regulated by BCL2 family proteins. They control the release of cytochrome C and other mitochondrial proteins into the cytosol. There are two classes: anti-apoptotic proteins, which inhibits apoptosis by blocking release, and pro-apoptotic proteins, which promotes apoptosis by enhancing release. The two subclasses of pro-apoptotic proteins are effector proteins, whose main proteins are BAX and BAC, and BH3 only proteins, which is the largest subclass of BCL2 family proteins. BH3, the BH domain shared by all classes, is what mediates the interactions between the two classes of BCL2 family proteins. These diagrams show how BCL2 family proteins release or inhibit the release of mitochondrial intermembrane proteins. In the absence of apoptotic stimulus, anti-apoptotic proteins bind to effector proteins, keeping them inactivated, therefore blocking release and inhibiting apoptosis. Effector proteins are activated by the presence of apoptotic stimulus, so they're prompted to aggregate on the outer mitochondrial membrane and form oligomers that induce release and therefore promote apoptosis. Also activated by the presence of apoptotic stimulus are BH3 only proteins. They promote apoptosis by either binding to anti-apoptotic proteins or binding to effector proteins. A key takeaway is that the balance between these activities determines the occurrence of cell death by the intrinsic pathway of apoptosis.
Inhibitors of apoptosis, or IAP proteins, represent a group of negative regulators of both caspases and cell death. Most prominently, IAPs control ubiquitin-dependent signaling events that regulate activation of nuclear factor Kb and mitogen-activated protein kinase, or MAPK, pathways that in turn drive expression of genes important for inflammation, immunity, cell migration, and cell survival. In the absence of an apoptotic stimulus, IAPs prevent accidental apoptosis caused by the spontaneous activation of procaspase. The IAPs are located in the cytosol and bind to and inhibit any spontaneously activated caspase. When an apoptotic stimulus activates the intrinsic pathway, among the proteins released from the mitochondrial intermembrane space are anti-IAP proteins, which bind to and block the inhibitory activity of the IAPs. At the same time, the released cytochrome C triggers the assembly of apoptosomes, which can now activate a caspase cascade, leading to apoptosis. Many of the cellular processes controlled by IAPs are frequently deregulated in cancer and directly or indirectly contribute to disease initiation, tumor maintenance, and/or progression, making IAPs obvious targets for anti-cancer therapy. Animal cells need signals from other cells, not only to grow and proliferate, but also to survive. If deprived of such survival factors, cells activate their intracellular death program and die by apoptosis. This arrangement ensures that cells survive only when and where they are needed. Survival factors, just like mitogens and growth factors, usually bind to cell surface receptors. Binding activates signaling pathways that keep the death program suppressed. Survival factors can suppress apoptosis in various ways. For example, it can increase the production of anti-apoptotic BCL2 protein. This BCL2 protein is created in the nucleus after the survival factor is binded to a receptor on the plasma membrane. Another way is the survival factor inactivates the pro-apoptotic BH3 only BCL2 protein. This is where the survival factor binds to the receptor again, which then activates an AKT kinase, which then turns inactive BCL2 into active BCL2. Survival factor can also inactivate anti-IAPs by binding to a receptor that activates a MAP kinase, which then activates HID, which allows IAPs to be produced. A role of survival factors is to adjust the number of developing nerve cells to the amount of target tissue. In this example, more nerve cells are produced than can be supported by the limited amount of survival factors released by the target cells. Therefore, some cells receive an insufficient amount of survival factors to avoid apoptosis. This strategy of overproduction followed by culling ensures that all target cells are contacted by nerve cells and that the extra nerve cells are automatically eliminated.
Can you repeat?
Oh, go ahead. Oh, okay. I just wanted to say one regular aspect of apoptosis that we pretty much use every day would probably be just maintaining the balance between cell death and cell division. Just maintaining that balance so that there's not too much cell division, which could lead to like tumor growth and cancer, and that's not too much cell death. So just maintaining that balance, and that's where apoptosis plays a key role.
Yeah, I'm from Group 4. What are some ways apoptosis can be prevented?
Well, there are several different inhibitors of apoptosis. Um, Austin, would you like to like clarify some or provide some examples that prevent apoptosis?
Yeah. Um, oh, okay. Just go over the inhibitors again. Well, could you go to the next slide? I think it's on the next slide.
Sure.
Yeah. So the types of survival factors are activate by signal regulators, which then make BCL proteins, and or they can inactivate anti-IAPs, which um, lets IAPs be created, which blocks apoptosis. It really depends on whether or not you have the survival factors.
Okay, thank you.
Another example. Oh, yeah. I just wanted to add just another example of an inhibitory protein. I found was FLIP. It's not illustrated in any of these slides, but essentially it behaves the same way as an initiator caspase, but it lacks the key cysteine amino acid that is necessary for the cleavage of essential proteins found throughout the cell. And so that's just another example of an inhibitory protein that prevents apoptosis.
Thank you.
Yeah, no problem.
Okay, so we're group A and we will be presenting on cancer. Oh, can you hear me now? Okay, so we're group A and we will be presenting on cancer.
Okay, so in all malignant cancers, cells begin to divide without stopping and they spread into surrounding tissues. Normally, our cells grow and divide to form new cells when the body needs them to, but when cancer develops, this process breaks down and it becomes unregulated. So old or damaged cells that should die instead survive, and then new cells form when they aren't needed. And these extra cells can divide without stopping, and that leads to the formation of tumors. So cancer is a genetic disease, and what I would mean by that is that cancers involve many individual mistakes and mutations in several specific genes within the cell, and these genes are called the proto-oncogenes. And when they mutate, it can be pretty bad. For example, with the right mutation, it can lose its ability to undergo apoptosis, or it can hide from immune cells, or it can multiply more quickly than normal. And these proto-oncogenes have an antagonist, and these are called the tumor suppressor genes. And they function to prevent mutations from happening by repairing DNA, or they order the cell to commit suicide if it thinks the cell is beyond repair. So tumor suppressor genes were first discovered when studying retinoblastoma. Retinoblastoma is an eye cancer that starts in the retina and is most commonly found in children. So if you look at the picture on the left, you can tell when someone has retinoblastoma. When you do flash photography, the pupils can appear white. And they found that afflicted children contained a deletion in their 13th chromosome, and they later isolated that gene within the 13th chromosome and found that it was the RB protein, the RB gene. And additional studies have found that the RB protein that is translated from the RB gene is essential in blocking inappropriate progression through restriction point. So it acts very much like the checkpoint. And within that same pathway, they found another tumor suppressor gene, which is the P16 gene. So if you look at the picture on the right, you can see both of them. So both of these genes are present in many tumors. So what this pathway shows is that if there is an active P16 and RB gene, then it inhibits the expression of S phase genes. And the S phase genes are primarily where DNA is replicated in the cell cycle. So if there are mutations in the P16 or RB genes, or if they're inactivated, then there can be no regulation of proliferation in that particular cell.
So what is P53? P53 is often called the guardian of the genome because its role in conserving stability by preventing genome mutations. P53 is a gene that makes a protein called tumor protein P53, also known as TP53, found in the nucleus. The functions include regulation of the cell cycle, apoptosis, and DNA repair. It helps keep abnormal cells, including cancer cells, from growing. P53 prevents neoplastic transformation either by cell cycle arrest or by triggering apoptosis. So the way it functions is, so let's say DNA damages a cell, which can be caused by agents such as toxic chemicals, radiation, or UV rays from the sun, which would lead to the triggering of the P53 gene, meaning there's an increased level of P53, which prevents cells from entering into S phase, leaving the cells in the G1 phase, which allows DNA to repair if the damage is not too much, which would then just continue the cell cycle. But if the DNA is damaged beyond repair, it will trigger apoptosis. The way it stops at the G1 is by P53 pauses the cell cycle by activating WAF1, which is also called P21, and P21 blocks CDK4 and cyclin D complex because the retinoblastoma protein cannot be phosphorylated, causing a stop at the G1 checkpoint. So how does P53 contribute to cancer? So the importance of P53 is to make sure that the damaged DNAs do not continue to proliferate. So if P53 is inhibited from either loss of alleles or mutations, the line of defense against stopping damaged DNA is unregulated. Mutations in the P53 gene may cause cancer cells to grow and spread in the body. Over 50% of human cancers carry loss of functions in P53 genes. An example of this is Li-Fraumeni syndrome. Li-Fraumeni syndrome is a rare disorder that greatly increases the risk of developing several types of cancer, particularly in children and young adults. Cancers most often associated with Li-Fraumeni syndrome include breast cancer, brain tumors, cancers of blood-forming tissues like leukemias, and a cancer called adrenocortical carcinoma that affects the outer layer of the adrenal glands. Nearly 3/4 of family with Li-Fraumeni syndrome and about 1/4 with Li-Fraumeni-like syndromes have germline mutations in the TP53 gene. Germline mutations are typically inherited and are present in essentially every cell in the body. Many of the mutations associated with Li-Fraumeni syndrome are changes in single amino acids in the part of the P53 protein that bind to DNA. Other mutations delete small amounts of DNA from that gene. These mutations result in an altered P53 protein that cannot regulate cell proliferation effectively and is unable to trigger apoptosis in cells with mutated or damaged DNA.
So as mentioned before, another driver of cancer involves proto-oncogenes. Proto-oncogenes are normal genes which regulate cell growth, and their functions include cell signaling pathways, cell survival, and growth controlling proteins that stimulate cell division, inhibit cell differentiation, and inhibit apoptosis. So proto-oncogenes lead to cancer when they undergo genetic changes and mutations and convert to oncogenes. Oncogenes continuously activate growth-promoting signals, leading to the development of cancer. And an example of this will be the RAS gene. So in a normal cell, the RAS gene will be activated by a growth factor receptor, which triggers intracellular phosphorylation and transcription to produce cyclin and CDK, which are drivers of the cell cycle. And when a hyperactivating mutation occurs, this activation process will occur continuously because of mutations that prevent GTP hydrolysis. The prevention of GTP hydrolysis will lock RAS in an active state and cause the overproduction of cyclin and CDK for cell growth.
How does cancer develop and progress? In a human body with more than 100 trillion cells, 1 billion experience mutations every day. If a mutation has selective advantage, that cell becomes a founder of a growing mutation. Mutations gain selective advantage due to the heterogeneity and the unchecked proliferation of tumor cells. Cancer is given a selective advantage not only over non-cancerous cells, but also against selective pressures that choose against it, such as pharmaceutical and clinical therapies, and also the host immune system. Reproduce without restraint and colonize foreign tissues. They also reproduce in defiance of the normal restraints on the cell cycle and invade and colonize territories normally reserved for other cells. If proliferation is too much, it can lead to tumor or neoplastic transformation. Cancer development is gradual and occurs in stages. A good example of this is cancer of the epithelium of the uterine cervix by the picture below. The picture shows how normal epithelium confined dividing cells to the basal layer, which is shown in A. B shows how low-grade epithelial neoplasia shows that the dividing cells are not confined to the basal layer anymore, but can be found throughout the lower third of the epithelium. And C shows high-grade intraepithelium neoplasia, which shows that the cell in all epithelial layers are proliferating and show no sign of differentiation. Differentiation. And then D shows when it's reached stage 4, where it's true malignancy, shows how the cells move through and destroy the basal lamina and invade connective tissues. So tumor progression. Tumors progress in rounds of mutations and proliferation using selective advantage because they grow without restraint and gives rise to fully malignant cancer cells. The picture shows how at each step, a single cell undergoes a mutation that either enhances cell proliferation or decreases cell death so it can become the dominant clone in the tumor. Proliferation of each clone keeps progression of tumor going, leading to more potential mutations. And then the final, where it says dangerous cell proliferation, it shows how the invasion throughout the basement membrane, which is an initial step of the metastasis. Metastasis is also where the means that cancer spreads to different parts of the body, where also known as stage 4. Not all tumors are as dangerous as malignant tumors, like for example, like benign tumors. Benign tumors remain inside the basal lamina that marks the boundary of normal structures compared to a malignant tumor that breaks the basal lamina and spreads. Although most benign tumors are non-life threatening, many types of benign tumors have the potential to become malignant through tumor progression. For this reason, other possible negative health effects, some benign tumors are removed by surgery. Cancer growth often depends on defective control of cell cycle, cell death, or cell differentiation. During the development of cancer, can either increase in cell division or inhibition of apoptosis can lead to the increased cell numbers, important for tumor genesis. They inhibit apoptosis by changing the function of anti or apoptotic proteins through post-translational modifications such as phosphorylation. Also contributing factors are that cancer cells are self-sufficient for their growth and proliferation, so they don't need a substrate. They are insensitive to anti-proliferative extracellular signals. They induce help from the normal stromal cells, and they produce telomerase or acquire another way of stabilizing their own telomerase.
So in a tumor cell, there are genetic and epigenetic changes observed. Epigenetic is defined as the genetic control by epigenetic is defined as the genetic control by factors other than an individual's DNA sequence. So changes in the epigenetic pathway can lead to abnormal gene activity and cause cancer. The three main epigenetic changes are DNA methylation, histone modification, and RNA associated silencing. RNA associated silencing involves genes that can be turned off by RNA when it is in the non-coding RNA form. And RNA can affect gene expression by causing heterochromatins to form or trigger the two other epigenetic changes. So the second epigenetic change that can happen involves histone modification. So histones are proteins that aid in condensing DNA into chromatin. The two main ways in which histones can be modified are through methylation and acetylation. When histones are acetylated, the chromatin takes a form known as euchromatin, where transcription factors can access the DNA and allow genes to be expressed. However, when histones are deacetylated, the chromatin becomes heterochromatin, which is an inactive form. And as you can kind of see in the image, the highly condensed nature of heterochromatin blocks transcription factor axis and gene expression, which can alter the transcription of genes that control the cell cycle. And the third epigenetic change is DNA methylation, which is the attachment of methyl groups to segments of DNA. The addition of methyl groups reinforces many gene silencing events, which can cause normal cells to become cancer cells. So, in a normal cell, these CPG islands are not methylated, but in a cancer cell, we often see the hypermethylation of CPG islands, which undo the work of tumor suppressors gene. This image kind of shows how an inactive gene can bypass checkpoints and continue to proliferate because of the hypermethylation. Hypermethylation can also lead to the instability of microsatellites, which are repeated sequences of DNA. And this instability can be related to a defect in the ability to correct mistakes that occur when DNA is copied in a cell, which can eventually lead to cancer. So, as a summary, cancer is unregulated cell proliferation that gradually develops. It is caused by two main changes to the cell: tumor suppressor genes, which inhibit cell proliferation, and the conversion of proto-oncogenes to oncogenes, which activate growth promoting signals. The three main epigenetic changes that can occur in tumors are RNA associated silencing, histone modification, and DNA methylation. And that concludes our presentation. Are there any questions? Or no? Yes.
So you're asking like what's the main reason for cancer growth? Mhm. Well, I think it's because there's unregulated growth of it. So like the whole point of it was like from when I was like talking about P53 was that it's supposed to stop the damaged DNA from proliferating. But if there is no P53 to keep that regulated, it just keeps continuing this damaged DNA. And well, the thing is about like cancer, it like benign isn't very harmful because it's like contained within the basal lamina. But as soon as it spreads out and it breaks through the basal lamina, it becomes like toxic, and that's like what cancer kills us. So I think like the main thing that continues like the worsening of cancer would probably be like no like stopping of that. So it just keeps proliferating at an alarming rate and just spreads throughout your body. When I was doing research, it's one of the things I think it can contribute to it is that this says by not checking a few if you let like some damaged mutated what's it called cells continue to grow, it leads to more of an opportunity for those damaged DNAs to continue on adding more mutations to it. So by not controlling it, the already damaged DNA has more potential for doing damage through other mutations that can come through it. Is what I'm trying to say. Do you get what I mean? Would selective advantage play into that? Okay. Mm-hmm. Okay.
I have a question. So I'm from group 3. So basically, going back to the topic of proto-oncogenes and oncogenes, can you briefly explain the mechanism by which, like the cell bypasses all of the checkpoints mentioned in previous groups and leads to like the rapid cell proliferation?
So, like the rash gene example, or is that what you want me to explain again?
Right. So like what basically causes the cell to the cancer cell to ignore all of the checkpoints in the cell cycle?
So the cells, so for proto-oncogenes, there are changed or they convert into oncogenes through mutations, and then these kind of bypass the checkpoints because this when the rash gene for this example is activated, so like it normally would like bind and then trigger intracellular phosphorylation, and then it would create the cyclin and CDK, right? But then for the mutation, when the mutation occurs, then this rash gene is continuously activated because of what in lecture says of the GTP hydrolysis. Is kind of locked, and then the rash gene is locked in the active state, and it causes the overproduction of cyclin and CDK. And then this kind of just bypasses the checkpoints uh, because let me see.
Actually, I think I kind of get it right now. So, because the RAS gene is activated and it leads to the overproduction of cyclin and CDK, I from the other group slides, isn't cyclin and CDK basically I guess like the lock-in key to it, like going through the cell cycle? Like if it's made and if like cyclin CDK is like phosphorylated or something, then it passes on like through the checkpoint.
Yeah, so then cyclin and CDK are drivers of the cell cycle. So they are normally, I think what the previous group said, they said that CDK is in a higher concentration. So then when cyclin is continuously produced by the RAS gene, the cell cycle will continue to like start this, the cell cycle will continuously to like start and like create and like divide more cells.
I see. Okay, thank you.